Hi, I would like to thank the organizer for giving us the opportunity to present our study on SFRP2 and its contribution to the transdifferentiation of type 2 cells. In idiopathic pulmonary fibrosis, or IPF, the alveolar type 2 cells are gradually lost and replaced with metaplastic keratin 5 positive basal cells. The question that we have in the lab and we are addressing with different studies is what are the mechanisms that promote this transdifferentiation? So it is well known that when type 2 cells are in organoid culture with MRC5, and MRC5 are fetal human lung cells, then they maintain a robust expression of SPC and therefore maintain their type 2-ness. However, we have made the finding that when we culture these type 2 cells with adult human lung mesenchyme cells, there is a progressive loss of the expression of SPC and the acquiring of keratin 5 expression. So this is the principle for which we are going to study what is the mechanism in leading to the transdifferentiation of the type 2 cells. And in our laboratory, Jamin and Shokun have already demonstrated that this transdifferentiation is dependent of the loss of BMP signaling. So we were originally interested in the role of senescent cells in the pathology of IPF. So we irradiated MRC5 and used them as a feeder for the type 2 cells in the organoid system. So as expected, when we grew organoid with type 2 cells and MRC5, we have the traditional robust expression of SPC and very little to none keratin 5 expression. However, when the type 2 cells are now cultured with irradiated MRC5, we have loss of the SPC expression and a strong gain of keratin 5 expression. And these images truly mimic what we have seen before when the type 2 cells were cultured with adult human mesenchymal cells. So from these observations, we hypothesized that both the adult mesenchymal cells and the irradiated DNA damaged fibroblasts release common factors and most likely secreted factors that are promoting this transdifferentiation. So how did we go about determining these common factors? So first, what we perform bulk RNA sequencing in both irradiated MRC5, non-irradiated MRC5, and then human mesenchymal cells, and again, and the fetal MRC5. We perform the analysis of the transcriptome of these two data sets, and we compare the differentially expressed gene to establish the list of genes that were first commonly found in both lists. We focus our attention on the genes that were upregulated, and we also only retain for our analysis only genes that were encoding a secreted protein. We identified a list of different genes. Some of them are that were already known from our group, that were the genes encoding protein that are known as being BMP antagonists that Shokun and Jamin have been working on. We also identified CCL13, IL-13, MMP3, and MMP12. However, we focus our attention on these genes that were encoding protein related to the VIN signaling for his well-known role in maintaining type 2-ness in type 2 cells. So among all these VIN related genes, the one we were focusing on was SFRP2, and why? First of all, as I mentioned, SFRP2 has been well described as a Wnt modulator, but it's depending on the context. So SFRP2 has been shown to promote or inhibit the Wnt signaling either in epithelial or mesenchymal cells. It also has been shown to be a BMP, BMP signaling inhibitors. But most importantly for us, the relevance to IPF was strong when after Max analyzed this transcriptome analysis of mesenchymal cells from control and IPF lung patients. And you can see that there is a dramatic increase of SFRP2 expression in IPF patient. However, when we did we tried to localize the cells, these SFRP2 positive cells, to see if they could, by localization, we could determine their function. And they are throughout the lung with no specific localization. In addition, SFRP2 expression is TGF-beta dependent, and we measure the expression of SFRP2 in MRC5 that have been treated with TGF-beta, and we show a nice upregulation of the gene that was blocked when we cultured the cells with TGF-beta and TGF-beta inhibitor. This was the same effect when we irradiated MRC5, where we show a nice upregulation of SFRP2 compared to non-irradiated cells. And that increased expression of SFRP2 was also blocked by TGF-beta inhibitors. So in both our model, we could determine that in MRC5 cells, SFRP2 expression was TGF-beta dependent. But what does it do with the transdifferentiation? So to test whether SFRP2 contribute to the type 2 transdifferentiation, we prepare organoid with type 2 cells and MRC5, and we treat it with various concentration of SFRP2. We first measure the colony forming efficiency, and regardless of the concentration of SFRP2 that we use, we saw a reduction in the colony forming efficiency. However, when we look at the expression of SPC and keratin 5, these expressions were dose dependent. So when as we increase the concentration of SFRP2, we are losing the expression of SPC. But in contrast, we are gaining the expression of keratin 5. So these experiments indicate that SFRP2 can promote AT2 transference, but it's dose dependent. Conversely, we confirm that SFRP2 is necessary for the type 2 transdifferentiation. And to do this, we silence SFRP2 expression in adult mesenchymal cells. We also did the same experiment with irradiated MRC5 and obtained similar data. So again, when we co-culture type 2 cells with human mesenchymal cells, we have the transdifferentiation of the type 2 cells with loss of SPC and gain of keratin 5. But then when we co-culture those type 2 cells with mesenchymal cells in which we silence SFRP2, we protected the expression of SPC, and we didn't gain much of the keratin 5 expression. So if we look at the colony, and we counted how many colony were keratin 5 positive, all of the colony were keratin 5 positive in the human mesenchymal cells treated with the negative control for siRNA. But with SFRP2, all of them were SPC positive, and some colony were still having some K5 expression, but it was always co-expression between SPC and keratin 5. So what is the mechanism by which SFRP2 can induce this transdifferentiation? So as I mentioned, it has been studied that SFRP2 could be a BMP antagonist. So we wanted to see if it was the case here. Shokun and Jamin has been showing that when you culture human alveolar type 2 cells with human mesenchymal cells. In absence of BMP4, they have the robust of functional keratinocyte. But if you add BMP4 to the culture, then again, SPC expression was preserved, which pretty much mimic what we have seen also in our siRNA experiment. So what we did is we culture human type 2 cells in 2D matrigel. We add BMP4 and SFRP2, and we followed the expression of phosphoMAD 138. So what we saw was with no treatment, there is very few cells that express phosphoMAD 138. However, upon activation by BMP4, most of the cells were phosphoMAD 138 positive, and the treatment with SFRP2 did not change didn't significantly change the number of cells that were phosphoMAD 138, which indicated to us that SFRP2 did not block BMP signaling in the 2D culture. And then when we confirm in the organoid system where we have type 2 cells, MRC5, plus or minus BMP4 and plus or minus SFRP2, we conclude the same thing. So upon treatment with BMP4, we have expression of SPC, as you expect, treatment with SFRP2, keratinocyte expression, but then when we combine SFRP2 and BMP4, we maintain the SPC expression. And then therefore, we don't think that SFRP2 act in this system as a BMP antagonist. So what could be then the function? So the next one that we looked at was how does it work for type SFRP2 in the Wnt signaling pathway. So in summary, what we have is that a non-activate mesenchymal cells such as MRC5 co-culture with type 2 cells maintain expression of SPC and the cells stay type 2 cells. However, when you have an injury in the lung and there is secretion or presence of TGF-beta 1, then these mesenchymal cells respond to the TGF-beta 1 by becoming activated and secrete SFRP2. And our data have shown that SFRP2 was able to promote the transdifferentiation from type 2 cells into beta cells, in addition to the environment that Jamie has been in his study showing that in order to get this transdifferentiation, you need to be with high TGF-beta environment, low Wnt and low BMPs. So we have some very preliminary data that indicate that AXIN2 expression, and AXIN2 is a Wnt signaling pathway target genes that goes up in Wnt. When Wnt signaling is activated, AXIN2 goes up. So we selected that gene to see if SFRP2 was able to block Wnt signaling in type 2 cells. So again, this is very preliminary, and the AXIN2 expression is actually reduced in type 2 cells when they are isolated from an organoid. However, if we measure AXIN2 expression level in the type 2 cells in 2D culture, so in absence of mesenchymal cells, SFRP2 is not able to block the activation of AXIN2 induced by Wnt3a. In addition to that, we also know from some primary study that Max has conducted that mesenchymal cell-free type 2 organoid treated with SFRP2 do not lose their SPC expression or gain keratin 5 expression, indicating to us that SFRP2 doesn't act directly on type 2 cells, but may act may act on the mesenchymal cells to act then subsequently on type 2 cells. So in addition to that, what we know is that SFRP2 was able to promote Wnt signaling in the activated mesenchymal cells. So our current working hypothesis is that SFRP2 promote the release of factor or factors from the activated mesenchymal cells that will then induce the downregulation or the inhibition of the Wnt pathway, which will then allow the type 2 cells to become basal cells. And so this is our current hypothesis, and we are focusing on finding those factors. All this work wouldn't have been done without the lab, so I'm really thankful to Hal Chapman to let me pursue this question, as well as Max and Jamin for their collaboration and ongoing sharing data with me. And with this, I will be happy to entertain any question. Thank you.